Welcome to the Molecular Therapy Podcast. I'm Dr. Rory Bricker-Anthony, Scientific Editor of the Molecular Therapy family of journals. This episode features a conversation between the Editor-in-Chief of Molecular Therapy Nucleic Acids, Dr. Paloma Giangrande, and Normand Allaire, Head of Genomics at the Cystic Fibrosis Foundation Therapeutics Lab. They will discuss an article recently published in Molecular Therapy Nucleic Acids titled Upregulation of a CFTR mRNA isoform has therapeutic potential for the treatment of 3' CFTR PTC variants. But first, if you're looking for ways to expand your expertise in all things cell and gene therapy, the ASGCT Learning Center is your go-to spot for on-demand learning. Our new expert-led content collections are now available, with topics including manuscript writing and journal strategy, grant writing for early career researchers, and more. Explore our course catalog today at asgct.org/learning. And now, Paloma Giangrande and Normand Allaire.
Welcome to mTNA's podcast. Today, we're diving into a breakthrough that could change the lives of the final 10% of patients with cystic fibrosis, or CF. These are people with CF who have nonsense mutations or stop signs in their DNA. For years, these mutations, like the one that we'll hear about today, this W1282X mutation, have been a wall we couldn't climb because the body destroys the instructions or the protein encoding mRNAs before the protein can even be made through this process that's termed nonsense-mediated decay, or NMD. But our guest today has found a way to bypass those stop signs entirely by tapping into a hidden, naturally occurring version of the CFTR mRNA or protein. And I'm joined today by Normand Allaire. He's the lead author of this recent study in Molecular Therapy Nucleic Acids that's titled Upregulation of a CFTR mRNA isoform has therapeutic potential for the treatment of 3' CFTR PTC variants. Normand Allaire, welcome to the show. It's really great to have you.
Oh, thank you so much for the invitation, Paul. I'm really happy to be here.
Yeah, and I guess before we get into the weeds, maybe let's simplify the science a little for our listeners and for those who are not as familiar with this topic. My understanding is that most CF research tries to fix the full-length message, or mRNA, but you looked at this shorter, truncated message that you call the E22 trunk. Yeah. Can you tell us a little bit more about why you believe this little-known isoform is the key to solving the nonsense-mediated decay, or NMD, problem?
Yeah, it's a great question. And before I jump into that, I thought first I wanted to just say thank you to all the people living with CF and their families. You know, they're a real inspiration for the work that we do. And I think it might be good to start with a little bit of background on CF to help the audience understand kind of where we're coming from, and then maybe talk a little bit about the role of the lab in the work against CF.
Great.
All right. So just a few sentences on some quick context. So CF is a multi-organ disease caused by variants in the gene CFTR. And so CFTR is a gated ion channel that sits at the apical surface of many epithelial tissues, including lung, pancreas, and intestine. And its primary role is to transport chloride ions across the epithelial surface and maintain proper hydration of the mucus layer. And when CFTR is disrupted, chloride transport is reduced and the mucus layer becomes thick and viscous, and that attracts bacteria and viruses and triggers inflammation and cell damage. And so the interesting thing about CFTR, it's a very large gene. It's about 190 kilobases and 27 exons, so it's really big. And just to put that in context, the average gene is around 20 to 30 kb and around 8 to 10 exons. And there are over 4,000 variants that have been reported in the gene CFTR. And most of these arise from normal human genetic variation. But about 1,000 of these have been shown to cause CF. And these affect, as you were alluding to in the introduction, these affect protein trafficking as well as protein gating. So the maturation of the protein to the apical surface where it acts, as well as the gating, the opening and closing of the channel. And then additionally, there are a subset of variants that result in no protein at all. And to answer your question about the CFTR modulators, or provide some context for that. So today, about 95% of people with CF are approved for treatment with CFTR modulators. And these are drugs that work on the protein themselves. So they help chaperone the protein to the apical surface. So defects that affect that, they can be useful for that. And the second thing is they help that opening and closing of the channel. But roughly 5% have premature termination codons and other mutations or variants that cause loss of protein altogether. And so the foundation's mission is to find a cure for all people with CF. And so in this effort, they're funding research on genetic therapies that can to find a one-size-fits-all cure for CF. But in addition to that, and this is kind of where our research fits in, we're working on finding alternative strategies that might complement those methods. And so that's kind of where our story begins today. With that background, that's kind of where we start, is one of these alternative approaches.
Yeah. And so maybe let's dive a little bit deeper into why this like truncated mRNA and what that really means for the research that you're doing. Because my understanding is that most researchers view truncated mRNA as really as trash. This is not an mRNA that gets translated. It's a mistake. It's not made into a protein. But what made your team suspect that this truncated E22 variant was not only stable, but could actually make functional protein, and so was bypassing this nonsense-mediated decay pathway?
Yeah, that's a great place to start. And I agree with you 100%. It's genuinely counterintuitive, right?
Yeah.
And that's kind of what makes the story kind of fun and interesting, and hopefully useful. So our lab has a focus on read-through therapies. So just to give you some context about the experiments that led up to this. So the read-through therapies are drugs that can help promote read-through of a premature termination codon. And there are reports in the literature that some premature termination codons could have a secondary effect on splicing. And so that was of concern to the lab because that could undermine our effects of read-through at certain PTCs. So we had done an experiment where we created 5 isogenic lines, each harboring a high-frequency CFTR PTC. And we did isoform profiling on that, targeted mRNA sequencing of CFTR isoforms. And what was interesting was that we found a drop in coverage, a drop in those reads between exons 22 and 27. And this occurred with 2 PTCs, W1282X, as well as R1162X. And those occur in exons 22 and 23 and 22. Interestingly, the 3 upstream PTCs, we didn't see this. These are Y1222X, R553X, and G542X. That was the first hint that we were looking at something that was slightly unusual, that was something related to exon 22. We didn't quite know what it was, but it was that asymmetry was giving us a clue that there was something interesting. And so what we did was we followed that up with 3' race and long read sequencing, because we went and we were actively looking for some kind of a truncated mRNA isoform at that point. And what we found was an mRNA isoform that contains exons 1 through 22, and then it contained additional 140 base pairs of intron 22, which is also unusual. And then following that, we saw a series of non-reference adenosine residues. And when we aligned that to the human genome, what we saw was that those adenosine residues aligned with an alternative polyadenylation signal, an intron 22. So that was our first hint that we were looking at not a degradation product, but a truly cleaved and alternative polyadenylated mRNA. And so we looked at that sequence and predicted what the protein would be from that. And what we found was that the protein would have been 1,258 amino acids, plus an additional 8 amino acids, we call those alternative amino acids, from that intronic region. And so what was interesting about that protein is that it was very similar in size and domain composition to a protein called CFTR 1281. The CFTR 1281 contains all the major domains except the final NBD2. And that has been reported in the literature to be responsive to modulators. So those two pieces of information, one that we had a truncated mRNA that appeared to be stable for PTCs downstream of exon 22, and the other side of it was that could give rise to a protein that had partial function when treated with modulators, made us think that this might be something that was therapeutically actionable.
Yeah, and so you're saying that the mechanism that's allowing this is that, if I'm understanding this correctly, is the retention of this intron sequence partially. Yeah, go ahead.
Yeah.
Go ahead.
No. It's a good question. And it's so, the question about how can this evade NMD, right?
Yeah, exactly. What's the mechanism there?
What's the mechanism, right? And so, I think that you hit the nail on the head. That's a key mechanistic question. And the answer is elegant, and it's elegant in its simplicity, honestly. So nonsense-mediated decay is a process that cleans up truncated mRNAs in the cell. And this is important because naturally occurring truncated mRNAs and proteins exist, and if left unchecked, they would give rise to some cellular toxicity. So the cell has this program called nonsense-mediated decay, which surveils the mRNA and looks for exon junction complexes. And if it sees an exon junction complex downstream of a stop codon, that's a signal that mRNA should be degraded. So the exon junction, let me just take a step back. Exon junction complexes are proteins that are deposited on the exon-exon boundaries during splicing. In a normal transcript, those happen all upstream of the termination codon. But in PTCs, you get exon junction complexes deposited after the PTC. And so that's the signal for nonsense-mediated decay to clean up those mRNAs.
Okay,
but in our case, during pre-mRNA splicing, we're activating an alternative polyadenylation and cleavage signal. And that effectively removes all the downstream sequence, including all potential exon junction complexes or where they would sit.
That's fascinating. How common is this?
I'm so glad you asked that question, Paloma. This is actually, and I was hoping you were going to ask that question. I should say this. So this is something that's just fascinated me. So alternative polyadenylation and cleavage. When we first did this, we thought that this was something unique when we were first studying this. And so we had done an experiment where we looked at putative alternative polyadenylation signals across the CFTR gene. And surprisingly, we found 250 putative alternative polyadenylation signals in CFTR, right? Exactly. Like, wow, how does that happen? And so, with that piece of information, you would kind of predict that you should be getting almost no full-length CFTR. It should be truncated all over the place because they exist in all the introns. And so that was a big question, was how is the transcript remaining full-length transcript? How is that being maintained? And what is happening that, we don't have activation of all these APA sites or alternative polyadenylation signals? So it turns out to kind of understand that, you have to understand a process called telescripting. So telescripting is a process that actively suppresses alternative polyadenylation. It's a critical process in the cell, and it's been reported, but you don't see a lot of press on it. And so we looked into this a little bit. And so the way it works is during splicing, there's a protein called U1 SNRP, and U1 SNRP is one of the initial proteins involved in the spliceosome, and it binds the 5' splice site. And so that's the well-known function of U1 SNRP, and that's the beginning of the building of this very large splicing complex on the 5' splice site. The secondary effect of U1 SNRP is that it inhibits alternative polyadenylation in proximal alternative polyadenylation. So part of the natural machinery of splicing is to actively suppress alternative polyadenylation because those sequences are so similar and so dispersed across CFTR and other genes. So that's the mechanism by which that's inhibited.
Wow. No, I mean, that's fascinating. And I was not aware of that. I guess my next question there is this also present in other genes or mRNAs?
Yeah,
Is it specific to the CFTR mRNA?
it's not specific to CFTR mRNA, not specific to it's a great question. It's not specific to CFTR mRNA. And we've been focused on CFTR and we were surprised by the number. I don't
Hmm.
actually know what the frequency is, but I do know
Okay.
that it's something that happens widely across genes.
Okay.
Yeah,
So potentially a broad therapeutic potential there that one could tap into. And maybe with that, let's transition to talk a little bit more about the technology that you chose to use, which
sure.
is antisense oligonucleotides that have
Yeah.
been around for decades
Yep.
and have been clinically de-risked. So we have drugs in the clinic that are referred to as ASOs or antisense oligonucleotides.
Yep.
And the way that they work is that they tile across an intron. And in your case, you tiled obviously across this intron 22 to see whether you were getting the positive effect that you were after. So
Right.
can you explain and touch a little bit on how an ASO acts like this genetic conductor
Yeah.
to tell
Yeah.
the cell which version of the protein to make?
Yeah.
And then we can talk more about why ASOs in particular and not other type of technologies that are
Yeah.
out there.
Yeah. So the way the ASOs work, they're basically as you know, there are two buckets of ASOs, large buckets of ASOs. There are ASOs called gapmer ASOs that trigger binding of RNA and then trigger degradation of that RNA. And we don't want to do that. So we use something called a splice blocking ASO. And so a splice blocking ASO binds but doesn't create degradation of the target. So, where I was briefly talking about this process of telescripting binds the U1 SNRP that is responsible for telescripting binds the 5' splice site. And so what we did was we tiled ASOs across the 5' splice site as well as the 3' splice site with the idea that we wanted to inhibit splicing and also promote alternative polyadenylation.
Mhm.
So we're kind of acting in two ways. We're going across the intron exon junction on the splice donor
Mhm.
site and the splice acceptor site. And we tiled ASOs across both those sites. And then we measured the increase in the amount of CFTR truncated mRNA. And we picked ASOs that gave us our largest increase in CFTR mRNA. And on the 5' side, it's kind of a dual action. It's not only blocking splicing, but it's also de-repressing telescripting. So we're
Oh,
taking the
so you
brakes
found
off.
that the 5' site was the key site to target.
Yeah,
And
it's the five.
okay.
Yeah,
And
the 5' site is where we get most of our effect. The 3' site gives us a little boost.
got it.
And
So
that's,
and so your like a potential therapy would be to use in combination ASOs that would target both sides, correct? To make it more effective.
yeah, that's what we did because we were looking for like
Yeah.
maximum effect. And so what we did was we were trying to upregulate that truncated isoform. And so our original thinking was that if we were to block splicing, that we would promote the use of the APA site.
Yeah.
And that's true, but mechanistically, it's true for a couple of reasons. One is that we're physically blocking the splicing, and the other one is that we're also inhibiting telescripting on the 5' splice site.
Yeah.
So, and with that, we're getting essentially wild type levels of the E22 trunk mRNA, and that's the key feature. This E22 trunk mRNA is typically at about 10% wild type levels,
Hmm.
and we see this kind of universally across all tissues, all wild type tissues.
Hmm.
Um, And we're able to, in both in primary cells and immortalized cells, we're able to increase that up to about wild type levels by evading
Wow.
NMD.
Okay,
So we
essentially
have a large effect.
restore, yeah, restore the full expression there, basically.
Exactly.
Yeah,
Exactly.
that's pretty fascinating. Yeah, and so this is actually a slightly unique use of ASOs because you're
Yeah,
really doing this combination to get that maximal effect.
exactly.
Yeah,
Yeah.
that's really interesting. And so obviously, like we said before, ASOs have been, have seen success in diseases like spinal muscular atrophy. They're drugs that many, many people take and are
Yep.
very successful in the clinic. For this particular application, because I'm assuming you're thinking about the next 10 years, right? And
Hmm.
potentially developing this into a drug. But what do you see as the unique challenges of delivering these ASOs to the lungs of, patients with CF? Given that, as you were describing, CF presents with all this mucus in the lungs, and the lungs
Right.
is really the organ that's the primary sort of reason why patients with CF fare very poorly. I mean, I
Right.
know that CF impacts, and you mentioned this, other organs as well, but the lung is really critical.
Yeah.
And really, my understanding is that drug delivery to the lung has been a hurdle in this particular disease.
Yep.
So, do you envision delivering your therapeutic? Does it have to go through the airway? Can you deliver it systemically and still see a benefit? Like,
Yep.
what are your thoughts around there?
Yeah. So it's a great question. And the comparison to SMA, I think, is a really good comparison because I think that the two diseases are very different, as you kind of alluded to.
Hmm.
With CF, it's a notoriously difficult tissue to target for nucleic acid therapeutics. And CF only adds to the complexity of that. And that's because we have this thick, sticky mucus that effectively traps and sequesters the ASO, right? So
Okay.
that's one challenge. In addition, even if you can get through the mucus barrier, the negatively charged ASO have a really difficult time transversing the cell membrane. And so that's a second barrier. And then finally, if you do transduce the mucus and transduce the cell membrane, there's a tendency for these ASOs to get stuck in the endosome. And so they may never reach their target, which is in the nucleus. So these are all, real problems and, and somewhat specific to CF. In addition, because CF is a chronically inflamed tissue,
Mhm.
it tends to be high in nucleases.
Yep.
And so
Yeah.
that's an additional problem that we have. So I think that we're thinking that the preferred route of delivery right now is an inhaled version. And that creates one more challenge because in the lung, we know that the CF manifests itself in a very patchy lung epithelium that some areas are more well ventilated and some areas are more obstructed. And so trying to get a good uniform drug distribution in that heterogeneous tissue is really difficult. And it's quite different than what happens in SMA, where we're delivering intrathecally and you have more of a long-lasting effect.
Mhm.
So, I mean, there are a number of things there that make it more challenging. Um, I will say that these are all real challenges of delivering to the lung, and I think that a lot of genetic medicines are going to face some of these same things. And I don't think that they're insurmountable, but I do want to point out that they are unique to CF, and it's things that we're going to have to address, whether it be genetic medicines or ASOs or a variety of things. So I think you hit the nail on the head there. It's pretty different than other diseases like SMA.
Yeah, yeah, and delivery is no matter I think the disease is a big hurdle
Exactly.
regardless.
Exactly.
I guess, do you see, I'm sure you're aware of companies and pharma that have spent millions and billions of dollars in developing approaches for CF or lung
Hmm.
delivery in particular.
Yep.
And I think there's excitement around using lipid nanoparticles to do
Yep.
that. Do you see that as a potential solution when you're going through the airways? And do you see that as a potential solution, potentially if you go systemically and go the other way in
Yeah.
delivering these ASOs?
Yeah. Again, another great question, Paloma. So right now our approach is to try to use a naked ASO, modified
Mm-hmm.
but naked, and not using lipid nanoparticle. And so that's what we used in all our development experiments. So that was in immortalized cells as well as HBEs starting at undifferentiated state, going through differentiation. Right now, we're working on trying to get them through into fully differentiated HBE or human bronchial epithelial cultures at air-liquid interface. And those more kind of faithfully represent the lung epithelium. And we do see a reduced efficacy when we treat naked ASOs in that context. So we're in the process of trying increased dose, increased exposure, and trying lots of different things to try to improve that efficacy in those airway cultures that look more like a normal human airway. You know?
And in for future studies, like when you're moving to in vivo, what is your preferred animal model that will then that you think may translate this clinically more effectively?
Yeah, so it's a great question. And the animal models are, we know that there's a mouse animal model. And the challenge with any of the animal models is that because we're acting at the splice sites, we can't use an animal model that has a cDNA version of CFTR. So
Mm-hmm.
we need, at bare minimum, we need a model that at least contains some of that intronic sequence, which
Yeah.
there are very few. So there is a mouse model. I was just talking to a researcher at Case Western yesterday, and there is a mouse model that's available that has a humanized W12A2X CFTR
Mhm.
insertion, and that has the full gene insertion. So that's one model. Unfortunately, the mouse doesn't have very much of a lung phenotype. It has more of an
Mhm.
intestinal phenotype, so there'll be some challenges there. So I think that going into animal models is definitely the next. First, improving our transduction in primary cells is kind of the first barrier. And then maybe we have to go with lipid nanoparticles. Maybe we can do it with dosing. We don't know. Um, the next thing would be to try to find that animal model that gives us a good lung phenotype. And so
Mhm.
that's something we're going to need to work on.
Yeah, because I'm assuming the sequence that you're trying to target is not cross-species reactive. And
Yeah,
even if you were to make it, yeah, you want to create that exact mutation.
exactly. And
Yeah, yeah,
splicing is, as you know, is notoriously finicky
yeah.
and challenged with even just small changes can have large effects. In terms of animal models, I should note that the foundation lab does not do any work with animal models. So any kind of animal model would be done outside of the lab in conjunction with some kind of partner. So
Great. I guess the question about dosing frequency, do we still expect that duration of action that we're familiar with and, with other ASO drugs in the clinic, in particular, as you had mentioned previously in the setting of SMA?
So the dosing is likely to be different than in SMA, in that in the intrathecal delivery with SMA, the dosing is much longer lasting. And so we anticipate that dosing with an ASO to the lung would likely be on a more frequent basis and require redosing. And we don't know exactly what that would look like because we haven't been in those model systems yet, but that's kind of what we're anticipating at this point.
Going back to what you said, restoring wild type function. And this was the wild type function, even of that truncated protein. But
Yep.
if I remember correctly, you also showed in your study that you were thinking broader. And so, in terms of combination therapies, with your combining your ASO strategy potentially with existing modulators like
Right.
Trikafta, I believe,
Hmm.
was the name of that, and that restored about 25% of wild type function. Is this the
Right.
wild type full length protein that you were
Yeah.
referring to?
Yeah.
And how do you see that? And when you combine that to, as a combination therapy moving forward
Yeah.
into the clinic?
So I think that it's a really good question. When we say wild type function, we're talking about function, not the protein is obviously a truncated protein and truncated
Hmm.
mRNA. So The functional readout is, we measure it to compare it to wild type. And the reason why we do that is because that's kind of the metric they use, because function correlates very strongly with disease severity.
Mhm.
And so to kind of put some context on the numbers, in CF, 1% to 2% of function, people experience severe disease, and that includes thick mucus, frequent infections, and progressive lung damage. Around 10% function, people have a noticeably milder course. At around 20 to 30% or above, patients often have near normal lung function and a substantially different disease course. So 25% represents a meaningful threshold. In practical terms, like how that would potentially benefit a patient, better function means the mucus becomes less viscous, the airways are less obstructed, the patients may cough less, they have easier time breathing, and they have greater exercise tolerance. And hopefully that means that in addition, they would have fewer mucus plugs and the bacteria would be less likely to persist. So potentially it could translate into fewer pulmonary exacerbations and fewer hospitalizations. So these are all like forward-looking statements. We'd have to actually get into the clinics to test these, but based on the natural history and what we've seen with patients that have what we call these residual function mutations, those things are what we could kind of predict. Now, I want to be careful to set expectations appropriately. 25% is an average functional estimate, and it's not, as I said, because of the patchiness of the disease, it's not uniformly distributed across the lung. And structural damage from years of disease like bronchiectasis does not fully reverse. So even though we could achieve that, we would expect patients to still require ongoing care and management. And so what we're describing here, again, being careful not to overpromise here, is that it's not a cure, but possibly a meaningful change and shift from the relentless burden to something a little bit more manageable. And so
And
that's what,
one potentially could also think about, since CF is screened, I believe, neonatally, right?
yep,
And so patients could potentially start on this treatment very early on to prevent that lung damage,
right,
which might even make it easier to deliver the ASO, because by that point, like if you're starting at the beginning, these lungs have not yet accumulated all that mucus and all that inflammation that can
right,
make it even harder to deliver an ASO
right,
type drug. I see it as a really positive thing, especially if you could hit that robust effect and, even patchy, but I'm assuming if you go into a lung that's still somewhat healthy,
yep,
then you might be able to have a significant restoration
yeah,
and prevent the disease from, or definitely slowing down
yeah,
the disease quite significantly. So, very, very exciting. I want to take maybe just the last 10 minutes or so to go back to what we had kind of started at the beginning to discuss the future of this and the broad applicability of this approach. That, really this isoform upregulation approach that you're describing is very elegant, but very nuanced, and in a
yeah,
way, a novel strategy because of the
yep,
way that it's being applied. ASO technology has been out there for, as we said, decades, but the way you are applying it is quite unique and different. And so, do you see this being a blueprint for treating other genetic diseases also caused by premature stop codons? And where do you see some of the advantages of this approach, for example, compared to other types of RNA therapies, like, we alluded to earlier, like tRNA therapies that are being explored to tackle these similar type of diseases? So just a little bit more of your ideas and speculations in the future of this particular modality or therapeutic approach.
yeah. So, premature termination codons or variants are, they're difficult to address. This kind of approach would apply to 3' terminal PTCs. So it's definitely, as you allude to, it's kind of a niche of variants that it could address. But we do see this as a possibility at a conceptual standpoint for addressing other diseases. And so, one example that we addressed in the paper is this is Duchenne's muscular dystrophy as a particularly interesting candidate. And we know from this is that 3' terminal PTCs in dystrophin are associated with a milder clinical phenotype called Becker's muscular dystrophy.
Mhm.
In addition, there's been some research that suggests that mini dystrophins that are truncated on the 3' end have shown improvements in animal models. And so, whether there exists an APA site that could be modulated in dystrophin, you'd have to do that work to check that, and you'd have to do rigorous testing to show that that was
Mhm.
a possibility. But I think that at least in principle, that would be an example of where the logic could potentially transfer.
Mhm.
Again, it would have to be followed up with, rigorous experimentation.
Yeah, yeah. I mean, for me, the excitement is I don't know, for example, of all the PTCs, what percentages of those the 3' covers. Is it like
Right.
10%? Is it 20? And I see, as I mentioned, advantages of several modalities, targeting particular indications. Like, there's so tRNAs, there's a lot of movement now with tRNAs and tRNAs entering the clinic to
Yep.
target some of these premature stop codons
Yep.
in various indications.
Yep.
There is, again, delivery is a huge hurdle even for
Hmm.
delivering. These are long molecules. You cannot modify them with standard oligochemistries, and so to stabilize them, requires delivery vehicles, for example, like LNPs,
Yep.
to deliver them to the right tissues, and then. I guess stability of these molecules in vivo, potential off targets, which,
Yep.
it's always, a question. ASOs being de-risked clinically to me seems a very elegant way of taking care, or at least of a subset of these,
Yep.
PTCs. So maybe let's take just the last minutes of the podcast to if you can tell us a little bit more about what your future looks like with respect to your work. What is your perceived timeline or the biggest hurdle that you see right now towards moving some of this work into the clinic and into clinical trials? And is this an area that you're exploring at all or interested in? Yeah, so just maybe a little bit on that.
Sure. So I think that, this is what we published so far has been a proof of concept.
Hmm.
And so, I want to be careful about, again, over-promising here. What we've shown is in the laboratory, we can induce this, we can use ASOs to induce this truncated CFTR mRNA, and that leads to a truncated CFTR protein, and then we can capture about 25% wild type function. I think the next barrier is to work on differentiated cells that more reflect the lung epithelium. And so those experiments are ongoing right now, and we'll continue to try to get those numbers to be up into that 25% range, which we believe is a therapeutically meaningful percentage. We did touch on animal models. Animal models, as they are, the lab does not do any work with animal models in-house, so that would all be done through some kind of a partner. And so that would be the next obvious step, but that is, certainly a downstream effort. And we really need to get through that first HBEs at ALI, make sure that we get good efficacy before we moved into that.
Yeah, no, that's very exciting. Well, there's a lot of work that still needs to be done. So
It does,
you're
exactly.
gonna be busy for a while. Yeah, well, thank you very much. I want to just say a few words to wrap up. In general, I think it's rare to see a study that doesn't just show a moderate biological effect, but actually shows a 25%, which is a meaningful percent in restoration of function, especially in these primary lung cells. I think this is something that needs to really be voiced loudly because it's not trivial. And so, I see really this as a massive or really important signal for the CF community that we're still trying really hard to make advances in this space to help patients with CF. And
Right.
so, Norm, I want to really thank you for sharing your journey and vision with us. And I wish you best of luck with everything that you're doing. This was a really exciting study. Thank you.
Yeah. Thank you so much, Paloma. It was a pleasure speaking with you today. I really enjoyed it.
Likewise.